And that's because it's very tough. We look at the first electron micrograph there. That's a picture of this straw, expanded, high magnification. And what we're after are what are called the cell walls, which is that tough material which makes the straw stand up, stops it falling over. And the next picture along, we see that that is the cell wall of the plant, and it's actually quite a complex structure. It's not like sugar. It's composed lots of different polymers to give the plant its strength. So, first problem we're faced with is how do we get this soluble so that our enzymes can attack it? And we call that deconstruction, or I call it blowing up the plant cell wall. We've got to find some way of taking this tough straw, softening it up, breaking it up, so that our enzymes can get in and start to turn this into glucose. Now if you look at the bottom right-hand corner, that's our target. One of these complex polymers in that cell wall is called cellulose, and it's a strong fibre. It's like cotton. Cotton is made of cellulose. Okay? So it's a strong fibre, and it's made up of literally hundreds and hundreds of glucose molecules, all joined together in very long strands. So that's what we want our enzymes to hydrolyse into glucose. When we get that glucose, we can get the yeast and ferment it into our alcohol. And that is what the programme at Nottingham is all about, the one based at Sutton Bonington. We call it Lignocellulose to ethanol, and it's been abbreviated as LACE. Now you might be thinking, where did the A come from? Well, it was something we put in fortuitously because we didn't want it to be lice. And okay, you have Nottingham Lace, so we have an association with Nottingham. So it's called the LACE programme, and it's how do we turn the lignocellulose, that tough cell wall, into ethanol. And as I said, first thing we've got to do is soften it up. And we call that a pre-treatment. Now the way industry do that at the moment is to put the straw into a vat of acid and heat it at 140 degrees C. Not very environmentally friendly. We want to look at more energy efficient, more economic, more environmentally friendly ways of doing that. And the sort of things we're going to be looking at are the use of microwaves with our colleagues up at main campus in engineering, or extrusion, which is a food science technique for making things like Monster Munch. So once we've done the pre-treatment, once we've softened this stuff up, the next step is to add those enzymes to carry out the hydrolysis, to convert that into the sugar that we want. Now, where are we going to get those enzymes from? Well, out there in nature, there are organisms that can destroy this quite happily. They live on it. They're called fungi. It's the filamentous fungi, and we've got several of them in that picture on the right there. And you're familiar with these. If you walk through the wood, you'll see a dead tree with a fungus growing out of it. That fungus is secreting enzymes that are degrading this material, turning it into sugars. And it's using those sugars to live. That's its food. So we're going to find the best enzymes for making this into glucose. And we're doing that by going out worldwide and taking strains of yeast growing in really weird and wonderful places. The first they've sampled was actually the stubble field out the back of Sutton Bonington. But they're going to go a bit wider than that. And they're looking for enzymes that will do that digestion. Once we've got the sugar, the soup of sugars, we can add the yeast, the fermentation yeast, and turn it into ethanol. And here we also have a series of challenges that we're trying to solve. We need to get improved yeast strains. The yeast we use at the moment are designed to use this simple sugar, not the straw. We need, for petrol, much higher levels of ethanol. So we need yeast that can not only produce more ethanol, but are resistant to it. The yeast get drunk as well. You know, there's too much there, they will die. So we want higher ethanol yield and better ethanol tolerance in our yeast. Inhibitor resistance. When we take this straw and pre-treat it and turn it into a soup, it's not just glucose that goes into solution. There's a lot of other chemicals, many of which are toxic to that yeast that's going to do the fermentation. So we want to reduce that or find yeast that are resistant, that are no longer killed by those toxins. I said that there's a lot of sugars in here. It's not just glucose. In fact, glucose only represents about half of the sugar in there. There are lots of other different types of sugar, and we want to find yeast that will use them. So as well as using the glucose, we'll utilise the whole of the sugar that we can produce. And resilience. These yeast are going to be in fermenters the size of this building. They're going to be stirred around and shaken. We wouldn't like it, they don't. We've got to make them tough enough to survive. Okay. So that is the programme. That's how we're trying to convert straw into ethanol. And it's a multidisciplinary programme. It doesn't just involve Paul and myself. It involves somewhere in the region of 20 people within University of Nottingham, several of which are sitting at the back as well. Hello. And as well as this, we have a series of themes within the programme. And at this point, I'd like to hand over to Paul to talk to you about those themes.